Have you ever pondered the significance of studying microbes, those minute creatures invisible to the naked eye? Welcome to the fascinating realm of microbiology, where we delve into the world of these tiny organisms that are omnipresent and hold a colossal impact on our lives. Microbes, small as they may be, play a vital role across various fields, from biotechnology to medicine and environmental science. They're the unseen heroes in our daily lives, aiding in digestion, recycling nutrients, and even protecting us from harmful pathogens. Yet they can also be the villains, causing diseases and environmental damage. It's like an intricate dance of nature, with each microbe playing a unique role. Imagine the possibilities if we can harness their potential, or prevent the damage they can cause. Their size may be microscopic, but their influence is anything but. So to comprehend this intricate world, scientists use a method called the 5 IES. Let's explore this process further. The 5 I's of studying microbes are inoculation, incubation, isolation, inspection, and identification. These 5 stages form a systematic approach that allows scientists to delve deep into the microscopic world and understand the characteristics and behaviors of microbes. Let's start with inoculation. This is the initial stage where a sample containing the microorganism is introduced onto a medium that will support its growth. Think of it as planting a seed in a fertile soil. The seed is the microbe and the soil is the medium. Next up is incubation. After inoculation, the sample is placed in conditions that are ideal for the growth of the microbe. This can mean a specific temperature, level of moisture, or other environmental factors. It's like providing the right amount of sunlight and water for the seed to sprout. The third stage is isolation. Once the microbes have grown, they are separated into individual colonies. This is done to ensure that the sample being studied is pure or uncontaminated by other microbes. It's similar to carefully selecting a single sprout from the garden for further study. The fourth stage is inspection. Here, the grown colonies are examined visually. Scientists look at the size, color, shape, and other physical characteristics of the colonies. This is akin to a gardener observing the appearance of a plant to identify it. The final stage is identification. In this stage, the microbe is identified based on its biochemical characteristics and genetic makeup. This can involve a variety of tests and techniques, from simple chemical reactions to complex DNA sequencing. It's like a botanist confirming the species of the plant based on its genetic characteristics. These 5 stages, the 5 IES, are not just sequential steps in a process. They are interconnected parts of a holistic approach to studying microbes. Each stage builds upon the previous one, contributing to a comprehensive understanding of the microbe under study. This systematic approach allows scientists to study microbes in detail and understand their characteristics and behaviors. It's through this meticulous process that we continue to unravel the fascinating secrets of the microscopic world around us. Bacteria come in a variety of shapes and arrangements, each with unique implications. Let's delve into this microscopic world. On one hand, we have rod-shaped bacteria, known as bacilli, which are like mini cylinders. Then there are spherical bacteria, known as culchi, which resemble tiny balls. And finally, we have the spiral bacteria, or spirilla, that look like little corkscrews. Now, these bacterial shapes aren't just for show. They play a key role in how bacteria interact with their environment, and even how they can cause disease. But it's not just the shape. The arrangement of these bacteria matters too. Some bacteria prefer to hang out in chains, while others form clusters. These arrangements can provide valuable insights into their behavior and potential impact. For example, chains of spherical bacteria, known as streptococci, can cause illnesses like strep throat. By understanding the shape and arrangement of bacteria, scientists can better predict their behavior and interactions. The world of microbes becomes visible under the microscope, especially when we apply techniques like cell staining. To study these minuscule life forms, scientists use different types of microscopy. Light microscopy, for instance, uses visible light and a series of magnifying lenses to reveal the basic structure of a cell. It's like peeking into a secret world, hidden from the naked eye. However, to step even deeper into this world, scientists turn to electron microscopy. This technique uses a beam of electrons instead of light, magnifying images up to 1,000,000 times their original size. It's like having a superhero's vision, able to scrutinize even the tiniest of details. While microscopy brings us closer to the world of microbes, cell staining helps us distinguish one microbe from another. Just as an artist uses different colors to bring a painting to life, scientists use various stains to highlight different parts of a cell. One example is gram staining, a method that divides bacteria into two large groups, gram-positive and gram-negative. This staining method colors gram-positive bacteria purple and gram-negative bacteria pink, making it easier to identify them under a microscope. Through these techniques, we are not just observing microbes, we are also understanding their structure, their characteristics, and how they function. It's like being a detective, piecing together clues to solve a mystery. Through microscopy and cell staining, the invisible world of microbes comes to light, offering a wealth of information. When it comes to bacteria, not all are created equal. There are significant differences between gram-positive and gram-negative bacterial cells. Let's delve into the world of these minuscule life forms and discover how they differ. The key distinction lies in their cell wall structures. Gram-positive bacteria have a thick peptidoglycan layer that retains the crystal violet dye during the gram stain process, resulting in a blue or purple appearance under the microscope. On the other hand, gram-negative bacteria possess a thinner peptidoglycan layer sandwiched between an inner and an outer lipid membrane. This structure doesn't retain the violet dye, but instead takes up the counterstain, safranine, and appears red or pink. This isn't merely a matter of aesthetics, though. The structural differences have significant implications in the realm of medicine. The thick peptidoglycan layer of gram-positive bacteria makes them susceptible to certain types of antibiotics, such as penicillin, which target this specific layer. However, the outer membrane of gram-negative bacteria serves as an effective barrier, often rendering these antibiotics ineffective. Furthermore, gram-negative bacteria can be more dangerous due to the presence of endotoxins in their outer membrane, which can trigger strong immune responses. This is why gram-negative infections are often harder to treat and can lead to severe conditions like sepsis. Understanding these differences is crucial in the field of medicine, especially when it comes to treating bacterial infections. The study of microbes is a fascinating journey into a world unseen by the naked eye. We've explored the 5 eyes of studying microbes, delved into the intriguing shapes and arrangements of bacteria, and highlighted the essential role of microscopy and cell staining. We've also differentiated between gram-positive and gram-negative bacteria, each with its unique characteristics. By delving deep into the microscopic world, we unravel the mysteries of these tiny organisms and their significant impact on our world.